Okay, sup guys. So it's this bullshit again. Okay, so I get to waste another 30 minutes of my life to help you guys with your fucking bio problems. So maybe if you paid a bit more attention in class or actually read the book, you wouldn't need this fucking goddamn service. But anyways, let's go over animal development and regulations of gene expression. Okay, like usual, I'm just going to go through the slides and then just basically explain the slides, blah blah blah. Okay. So, what we're probably going to first talk about is chapter 47, which was just basically the animal development. So, as you can see, it talks about how animal development occurs at all stages of the life cycle. And that's important because without development, you would still be a blob of cells, basically. And that would suck because you wouldn't be able to do anything really as a blob of cells. And to deal with that stuff, you have to go for metamorphosis and gamete production, as well as embryonic development. And so basically when they're talking about that, those are the main things that are involved with the cell division within before a fetus is formed, basically. It's like, yeah, it's still a fetus, basically. And then animals display different body patterns. They share many basic developments of blah blah blah. So basically, they have genes that tell them how to set up their bodies. And then biologists use model organisms, which are just basically organisms that are easy to study. And one of them would be the frog. And as you can see, here is the embryonic development part, and that is the important part. And we're supposed to take notes in class about this, but a lot of you probably still don't know what the hell this is. So let's go over this in detail. Okay, so the first part, as you saw earlier, was fertilization. And fertilization is basically just where the sperm and egg come together. If you didn't fucking know that, I don't know how to help you, because that's what we've been doing in biology classes since, I don't know, maybe even like before 6th grade. It's just, sad if you don't know that. So basically, the sperm penetrates the protective layer around the egg. It gets inside, it changes the inside of it, preventing polyspermy. If you want to look into more detail about that, you could look on the chapter 47 book slides. They have this beautiful complex thing about how it prevents it from going into like, oh, look at the Ca ions, like prevent it. It's like a cortical reaction, blah blah. Of course, that's too much detail. You don't actually really need to know that unless you're just like, you love learning. But I highly doubt you're one of those people because you're watching a review video instead of actually doing your work. And then, so yeah. And then cleavage. Cleavage is the next part. It's just basically when the cell starts dividing into becoming a blob. The blob is important because it's pretty much what you start out as a blob. So you need that blob. So it's just basically rapid cell growth, and it does not grow. Like, it just divides. It goes from one big cell to many small cells, but the small cells, like, it doesn't increase in volume, if you want to think of it that way. And then, yeah, it just partitions the cytoplasm of one large cell into smaller cells called blastomeres. So basically, each of these individual cells are called the blastomeres. And the solid ball of them, this whole thing, would be called the morula. Okay, excuse me for my pronunciations of probably shit, but yeah, sucks to suck. And then the bastula is the ball of cells, but then on the inside there's a fluid-filled cavity that's called the blastoa. And here's a picture of the blastoa within the morula or bastula. So here, blastoa, and then yeah. And there's also the vegetal pole and animal pole. How those are determined is a little bit more complicated. It is in the other slide if you if you care about that kind of crap. Basically, the amount of yolk on both sides is the determining factor, if I'm correct, on why. Damn, they have so much detail on this stuff. Yeah, basically the cleavage patterns are important. We didn't even talk about cleavage patterns, but basically how the yolk is distributed tells us where the vegetable pole and animal pole will happen. And you see the first cleavage makes it equal, but then the third one is asymmetric. and you see it's not symmetric, and eventually it makes a bastille near the animal pole. Of course, you don't need to know that shit, so, yep. Cool. Here's a picture of it, basically his actual cell. Here's a fertilized egg, it's one big cell, then the 4 cells, and the earlier bacille, you can see now there's a whole thing, and then a later bacille, now it's big. Yay! And now the next important part, which is morphogenesis. Okay, so basically after cleavage is done, cell division slows down, and the cell cycle is restored. Then what does that basically mean? So during morphogenesis, we have gastrulation and organogenesis. And if you want to go back to that diagram, gastrulation was this part and organogenesis was this part. So basically, gastrulation just rearranges the cells into a free ladder embryo. If you want to look at the diagram, they should have a diagram of it. Huh. That's really weird. Well, fuck. I guess I'll have to go look up a goddamn slide of it. Gastrulation. Okay, here. That's this. So this is the gastrule, as you can see. So now it's 3 layers. There's the ectoderm, the endoderm, and this is the. Where's the mesoderm? Okay, this stuff is a mesoderm. They just decide. Wait, what the fuck? That should be the mesoderm. What they call the blastilla. This is fucking retarded. Okay, let's get this. Mesoderm is the middle one, endoderm, and ectoderm. And then there's also the bacillor 4, and yeah. Cool. So this is the point where it becomes the free cell layers, basically. Yay. And it's called the gastrula. And then organogenesis, literally organ genesis, birth of organs. So it's the formation of organs. And then yeah, there's 3 layers that we're talking about. There's the ectoderm, and then to remember how the germ layers form what they do. Ectoderm, basically, it's on the outside, so obviously it makes sense that would make your skin and that kind of shit. And then there's the endoderm. Since endoderm is at the very middle, it kind of makes sense it would make your digestive tract, because that's like the most inside of you, or arguably still outside of you, because technically your stomach and whatnot is not technically the inside, it's actually still technically the outside because it's connected with your mouth and whatnot, but ignoring that, it is basically the most inside of it. And then we have the mesoderm, which is just basically the shit in between. So like your muscles, all that crap. Just everything else, basically. And yeah, here, if you want to memorize this shit, which you technically kind of do need to do if you want to take the SAT2 bio, because I've been practicing that shit, and yeah, you need to basically know that what they make, but in this class, apparently you don't really need to know, but yeah, cool. This just tells you the kind of shit it makes. Yay! Ah, fun. Do we want to watch the video? No, because we already watched the video. Yeah. So here's a picture of this kind of shit. So here we go. Here's the basoa. And then there's the goddamn, what is it called? So this is like when it's still just the, what is it called? Oh my god. Talk about me being bad at math science. Yeah, this is when it's still a blastula, basically. So this is when it's still a blastula, and then apparently they're called mesenchyme cells. I guess that kind of shit doesn't really matter, but basically they start moving around and moving and moving until we eventually get to the important part where it's actually becoming the free layers, the free germ layers, I think they're called. Yeah, the free germ layers. Yay. Okay, so yep. So they're moving around, they're moving around, and now you get to the free layers. Ectoderm, mesoderm, endoderm. Yay. Yay. Who the fuck gives a shit? I don't know. And then we have organogenesis, which is just basically it's basically where the various regions of the germ layers become rudimentary organs. Yay. So basically, your cells are specializing into what they're supposed to be. Yay. And in early vertebral organogenesis, the notochord forms from the mesoderm and the neural plates form the ectoderm. Oh, great. Great to know. You see, look. This is the this is the ectoderm, and here it's becoming nerves. You see, it's folding inwards, and then this becomes a neural tube. Oh my goodness, what the fuck? Yay! Who gives a shit? I don't know either. We want to watch these videos? Not really, and that's not important. Not important. No, oh, well, technically this shit is important. Remember that we have a quiz tomorrow. Yeah. And then this shit not important, and this oh yes, this shit is important. So then there's program of differential gene expression leads to different cell types in a multicellular organism. What does that even fucking mean? It basically is telling you your genes tell your cells, tells your very first cell, your fertilized egg, into being tell it what to become. So yeah, during embryonic development, the fertilized egg gives rise to many different cell types. What does that mean? It's just basically you start out with one cell, that egg cell, and then it differentiates and differentiates, and then all of a sudden, look, you have neurons, you have your epithelial tissue on the inside of your stomach, you have your epithelial tissue in your nasal cavity, you have the tissue, you have retinal tissue, you have what? You have, well, retinal tissue is actually kind of wrong to say. You have your muscles, you have your skeletal, it's like everything. It makes your connective tissue, blah. And it's all different, but it all came from one egg. Now, isn't that amazing? So yeah, cell types organize successively into tissues, organs, organ systems, a whole organism. You should know that. We've talked about that in 7th grade bio as well. And then gene expression orchestrates the development of this. And this is the important part. Genes are what tell your body to do this. And then there's the sequential regulation of gene expression during cellular differentiation. And this is basically the shit we were talking about in class. Determination is just all the crap before differentiation. It's like your body preparing to differentiate. So it's like it's turning on the correct genes that will like tell your body to differentiate like into an arm in one area and into a leg in another area, blah blah, all that crap. And the differentiation is the final part where yay, you actually do what you're supposed to be fucking doing. So the cell that was told to do what they're supposed to do is now doing it, basically. Yay. Differential gene expression leads to different cell types in a multicellular organism. Okay, basically, different genes are expressed in different areas, and obviously, genes tell your cell what to do, so they do different things and become different types of cells in your body. And then the zygote undergoes transformation for three interrelated processes: cell division, morphogenesis, and cell differentiation. Wow, I did not know that. Totally did not know. So basically, cell division, what is that? Mitosis increases the number of cells. Duh. That's what we've been doing earlier. And then morphogenesis is just basically your cells moving around, differentiating, blah, 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 and becoming, well, not differentiating, so to speak, but it's like moving, your cells are moving together, making tissues and whatnot, which will eventually become organisms, would then become organism systems, which then become the whole organism. And if you want to go into ecology, we can also talk about populations and whatnot, but that's not very important right now because we're not doing ecology. Then we can talk about cell differentiation, and then cells become specialized into structure and function. Yeah. So basically, important. Genes tell your cell to do something, they do it, and that's what makes your nerve cell different from your skin cell, even though they're technically all the same DNA. Okay, here's a diagram of this crap again. Here's the zygote, yay, egg cells, and there it's like, what is it? It's become the... Wow, I'm so fucking bad. What is the ball of cells called? It's so... Shit, I'm so bad. Yeah, it's called a Marala. Oh my god. So remember that's a Marala. Don't be like me and be a fuckboy and not remember what the fuck it is. This is the Marala. And then comes the Blastula with the... What is it? Blast... Blastiol. I can't pronounce it. It's like Blastiol. Blah. It's the hole in the middle. Just remember that. And it's kind of related to Blastiol. And yeah, and then the ectoderm, it's beginning to make the free layers, and those free layers will eventually become the adult sea star. Yay! And here's a plant version of it as well. It's whatever. It's like same idea basically. And this is just basically it goes from a frog, from an egg, to a tadpole. Oh my goodness. This shit's not very important. Okay. Apoptosis. Yep. We all heard about this one. At least you should have heard about it. It's programmed cell death, controlled cell death, whatever you want to call it. It's just basically when your body tells your cell to die, instead of your cell dying uncontrollably, which would be called necrosis. Necrosis is usually worse than apoptosis because it's uncontrolled. And okay, at various times during development, individual cells, sets of cells, a whole tissue stop developing or engulfed by neighboring cells. Cool. So basically, yeah. Basically, you make too many of one cell, your body then realizes you made too many, and it just kills them off with apoptosis. Done. It's like, oh, your neurons? You made too many neurons when you were a child, so they just kill them all. Although, some of you look like you actually needed those extra neurons. Yeah, truly disappointing. Yeah, I'm just kidding. Don't take it seriously what I'm saying. And then another example is morphogenesis and the fingers and toes. Basically, you have apoptosis that kills off the webbing in your hands. Because we used to be fish, like a long time ago, our ancestors were the fish. We needed webbing. Eventually, we didn't need that webbing anymore because, webbing doesn't really help when you're trying to write with a pencil. It kind of is more helpful while swimming. I mean, yeah, when we're swimming, it's helpful, but most of the time we're not swimming, so yeah. Yep, and then this is just basically talking about how we have the suicide built-in suicide mechanism to do important things. So basically, the basic mechanism involved in early animal development. Oh, and then timely activation of apoptosis proteins in some cells function during normal cell development, blah, blah. We can go back to the nervous system thing. It's like, oh, your body kills off certain cells because they're not, like, you made too many of them, and, like, it would be bad to have so many. Your immune system, actually, majority of your immune system cells will die before they even get to have a job because they fail to show that they can control which cells that they want to kill, and when that happens they just get killed off. If that does not happen and they get released, that's when they start attacking your own tissue, and that would be called a... Wow, it's called an autoimmune disease. There we go. It's called an autoimmune disease, but that's not important. Blah blah blah. Yeah. Yep, and then if cell suicide doesn't happen, it can create problems like you might still have webbed fingers and toes. Although that can be fixed with surgery these days. They just basically cut it off while you're a child. But yeah, you can have webbed fingers and toes, and certain degenerative diseases happen from not having all your nervous system activating apoptosis. And then of course, the one that we all know is like apoptosis of cancer cells is important, and when apoptosis fails we get cancers, and when we get cancers we get sick, and then when we get sick we possibly get better, or more likely we die. Yeah. Cytoplasmic determinants and inductive signals. Okay then, basically the cytoplastic determinants are shit from your mother that then tells the egg cell on what it's supposed to do. So basically, It's just your mother giving some proteins and whatnot, RNA, blah blah. I mean, I don't think it's actually RNA. I think it's just some proteins. It just basically informs your cell what the hell it's supposed to be doing. It like because your egg cell is not going to initially know everything that's supposed to do, although it'll have quite a bit of information. But so the cytoplasmic determinants help push it along basically, and they came from your mother. And as the IGO determines, cells contain different cytoplasmic determinants, which lead to different gene expression. Yay. So basically, the cytoplasmic determinants, when your cell divides, some of them go over here, some of them go over there, and that determines what each cell will become into eventually. So yeah, look, here's the image. They have the two different things. They have the circles and the triangles. Of course, they don't look like circles and they don't actually look like triangles, but it's a nice representation, so why not? Yeah, so basically, your cell divides, and then a lot of them get into one area, and a lot go into the other area, and when it does that, it will eventually tell them which one should become, let's say, your head, and which one will become your leg, so to speak. And then another important source of information is from the environment around the cell, and yeah, especially signals from nearby embryonic cells. In the process called induction, signal molecules from nearby cells cause transcriptional changes in nearby target cells. Fun. So basically, the egg cells, once it's divided into multiple cells, the blastomere of each one blastomere might tell the other blastomere to become a nerve cell, and another blastomere might tell that blastomere to become like a muscle cell, and then like all of them tell each other what they're supposed to become, and then yeah, you have that organism. So their interactions between each other help to specialize each cell type. You see? Look at it. So here's those blastomeres at the bottom. They're sending signals out. Wow, look at it. It's cell signaling, guys. Oh my goodness. Don't you love cell signaling? It's cell signaling, so it sends out the signal molecule. It goes to the signal receptor, and then it's the transduction pathway to the nucleus. Oh my goodness, it all connects, guys. But yeah, basically that's it. Oh shit, this is such a waste of my time. Oh, look at that. Isn't this like from chapter 11? Don't you remember this? It was absolutely beautiful. Okay, but anyways. And then pattern formation, setting up the body plan. So yeah, pattern formation is development of spatial organization of tissues and organs. So basically, literally, it's giving you a pattern for the organism to follow, so it makes the correct tissues in the right area and the correct organs in the right area along the major axes. Yay! They begin on the major axes. Wow! Isn't that amazing? And the positional information, the molecular cues that control pattern formation, tell a cell its location relative to the body axis to nadir cells. So basically, it sends out those molecules from each cell, and they're telling each other what the hell they're supposed to be doing, and it gives those other cells a kind of general idea of where they are and what they're going to be. Okay, and these are famous people. I mean, they're cool and all, but who gives a shit about the history? The important part is the homeogenic genes, which are just basically there's these genes that tell your cell what they're supposed to be and where they're supposed to be. So basically, there's a gene that'll tell you, oh, you're going to become a nerve cell, and you're going to be found in the brain. But then if the gene mutates and it gets fucked up, it might like tell it, oh, you're supposed to be in I don't know, in your foot. A nerve cell that's supposed to be for your brain is in your foot. That's not helpful. That's going to kill you most likely. But yeah. So basically gene mutations of these will really fuck you up. But yeah. So basically they give you a guideline of what they're supposed to do. And then pattern formation is the development of a spatial organization of tissues and organs. Yay, we kind of basically get the idea. It's just you're in spatial, like in space, certain areas will be like your front torso will become, you know, the organs are in your anterior section of your body, and then your posterior section of your body make different organs. Like, I don't know, posterior tissues will become like your kidneys and whatnot because those are more to the back, and then your anterior tissues might become uh so many things. I don't know. Maybe you're, yeah. I have no idea. I really don't give a shit. But basically, in space, it's going to tell them what they're going to do. And then positional information is the molecular cues that control pattern formation. So basically, positional information are the small molecules that eventually will tell how the pattern formation is supposed to be. And it does this through cells interacting with each other. Whoops, I went up. Wait, what? Oh, whoops, I repeated the same thing twice. I'm sorry, guys. That was a waste of my time. Cool. This is basically what happens when a homeotic gene mutates. This is supposed to be an antenna on the fly. This is what happens when it gets fucked up and now all of a sudden it's growing a leg on its antenna. That's not good for the fly. It's pretty bad. Yeah. Yeah. And then we have the, oh my goodness. These are anatomical planes. I haven't seen these since Honor's Anatomy. Oh my goodness. But anyways, yeah. So yeah, basically the body axes, it tells it, oh, put the head here, put the thorax here, put the abdomen here, blah, blah, blah. And here's basically from the cell how it's doing that. So there's the egg, and then the fertilization happens. Woo. And then basically it's going to the different ectoderm, endoderm, they're going to move around. They're going to specialize into each part that they're supposed to become. Maternal effect genes encode for the cytoplasmic determinants that initially subspecies of the body. There's cool. Basically, there's these genes that tell the fly where their axes are so that they know to put their head on the anterior portion and not on their ass, not on their posterior section. Yeah. And then the maternal genes are also called egg polarity genes because they control the orientation of the egg and consequently the fly. Cool. So basically, these are the same thing. Yay. Wow. This is truly amazing. Look at this. This is one cell on one side, and these are different types of cells on the other side to tell you what it's developing into, what to become. Wow. Amazing. And then we have bicoid genes, and the bicoid genes are just basically the ones that affect the front half. And what are the front half? Those would be the anterior portions. Yes. And then if they don't have functional bicoids, they lack the front half of their body. Wow. And they duplicate the posterior sectors at both ends. So basically, if it was like in a human, you'd have no kidneys, but you would have like crap. I can't think of anything near the front right now. I guess your cardiac tissue would be at your front. So maybe two hearts and shit like that. It would be bad. You would not survive that kind of shit. Okay, yeah. So here it is. It makes a head and a tail, but when it gets fucked up, it makes a tail and a tail. Yep, and so basically this is once again going back to the morphogenesis shit, and more specifically like the kind of like the cytoplasmic determinants. Basically, the gene is more concentrated at the portion that's supposed to be at the anterior end, and when it's more concentrated, it becomes the head. And then the morphogene gradient hypothesis, which basically the gradient of substances called morphogenes establishes embryos' access and other features. So basically, where things are. Oh shit! I poured water on myself. Fuck. Ah. Okay. Basically, it tells where everything is supposed to be when there's different substance. Okay, it's really fucking cold now. God damn. So yeah, basically different morphogenes go onto each side and they tell what the thing is supposed to become eventually. These are pictures of basically morphogenesis, the idea of it. So look, there's tons of bicoid in one area. And it'll become the head. Yay. Why is this important? Apparently it's because it identified a specific protein required for early steps in pattern formation. Cool, so basically it told them, bicoid basically told them, wow, this thing makes the head. And then increased understanding of the mother's role in embryonic development. So basically, yeah, it just tells them that the mother has a pretty important development in how the embryo becomes. And it demonstrated a key development principle that the gradients of molecules can determine the polarity and position of the embryo. So basically, having a lot of crap on one side and not a lot of crap on the other side will tell the cells to one end make the head and one end make the tail. Oh, and these were the questions were in class. If you all might just slightly go over them. The first diagram below shows the levels of mRNA, two genes, blah blah blah, are coddled are different positions along the anterior, posterior, blah blah blah, and before function. The second diagram shows the level of corresponding proteins along the anterior axis, blah 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 blah. So basically, there's tons of bicoid in the anterior post, and then there's just the caudal genes. They're just like everywhere from the anterior to posterior, but there's tons of anterior in one area. And then there's bicoid in the anterior, and then the location of the egg, and then there's more caudals go. Oh, wow, isn't that amazing? So basically, which is the best supported by the data? Is it A? Is it B? Is it C? Or is it D? Well, let me think about this. Bicoid protein inhibits translation of caudal mRNA. Yeah, kind of makes sense. Bicoid protein stabilizes. No. Where do you get that from? That doesn't make sense. It's just like randomly put there. Translation of bicoid mRNA produces caudal. Where did you get that from? How did you think bicoid became caudal protein? That makes no fucking sense. And then the final one. Shit, the stupid slide thing is in the way. Okay, caudal proteins stimulate development of anterior structures. No. What the fuck? What we've been just learning is the bicoid is what makes anterior. So, why would the caudal make the anterior? No. Even if you didn't, I wasn't 100% sure A was right. All these other free freaking things don't make any fucking sense. So the answer is A. Whew, and another one. And this one just has even more shit to deal with. So the diagram above shows the developing worm embryo, blah, 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 blah. You know what? I really don't want to read this, but I guess I'll read it. Okay, the diagram above shows a developing worm embryo at the 4 cell stages. Experiments have shown that when cell free divides, the anterior daughter cell gives rise to muscle, and the gonads and the posterior daughter cell gives rise to the intestines. However, the cells of the embryo are separate from one another. Early during the 4 cell stages, no intestines will form. Other experiments have shown that if the cell free and cell 4 are recombined at the initial separation, the posterior daughter cell of cell free will once again give rise to normal intestines. Which of the following is the most probable explanation for these findings? So basically, they have the 4 cells. And then if you pull them apart, but then put them back together, if you pull them apart, they notice that cell free, it gives rise to muscle and gonads, and the posterior daughter cell, cell free, gives rise to intestines. From just cell free. And then let's go through these. A cell protein on cell 4 signals cell free to induce formation of the worm's intestines. That does make slight sense. Because remember what we were talking about, how the cells communicate with each other through induction and whatnot? Yeah. So it kind of makes sense. Maybe that cell sent a signal out, oh, you need to become intestine, so go do that. So kind of makes sense. Plasma membrane of cell 4 interacts with plasma membrane of the posterior portion of cell 3, causing invaginations that become microvilli. What the fuck? Just microvilli are an important part of intestines, yes, but they're not the main thing. That's just a tiny part of the whole thing. So no, that doesn't give enough information for what it needs to become. So no, of course not. Cell 3 passes an electrical signal to cell 4, which induces differentiation in cell 4. Okay. Two pretty important things. This is going the wrong fucking way. This went from cell 3 to cell 4. That's wrong. We're talking about cell 4 to cell 3. What the fuck? That's wrong. You can just immediately throw it out. But even if it was from cell 4 to cell 3, no cell sends signals through electrical signaling. Because when a cell goes across another cell, there's not an electrical impulse that can jump across. Even your neurons, which are basically electrical signal based cells, don't do that. They have to send out neurotransmitters, which are chemicals, to then bridge a gap. They don't send the electrical signal from one side to the other. That's not how it works. And then cell 4 transfers genetic material to cell 3, which directs the development of intestinal cells. Okay. What the fuck? You want to tell me that the information in the nucleus in cell 4 right here somehow manages to get out of its nuclear envelope, get into the cytoplasm, and then get out of the cell itself, go into the intracellular region, and then go into the cytoplasm of cell 3 and into its nucleus. Is that what you want to tell me? No. That's not how it works. Your DNA doesn't travel out like that. Unless you had like necrosis where the cell spontaneously died and then the nucleus went everywhere and whatnot. And maybe a little might be able to get in, but no. That's not how it works. So obviously the answer is A. Don't need this shit. And well, this stuff is actually this stuff is technically tomorrow's shit, but if I remember correctly, this stuff was going to be on the test as well, so I'll just go over it. Determination is a term used to describe the process by which a cell or group of cells become committed to a particular fate. So basically, determination determines what its fate will be. Wow. And differentiation is a resulting specialization in structure and function. So basically, differentiate. You become different things. So it's specialized into different structures and different functions. Because structure determines function. That is a goddamn important thing. Structure and function are interrelated. So if it becomes a different structure, it becomes a different function as well. And the fate maps are diagrams showing the organs and other structures that arise from each region of the embryo. Cool. So basically, they show how the organs and other structures arise from each part of the embryo, which, like, remember what we were talking about on the blastoma, how the ectoderm becomes one part, mesoderm becomes another part, and endoderm becomes another part. Yay, that's basically what it is. Yay. Oh, look at that. We're doing it again. This becomes this part, and this becomes that part, and blah, blah, blah. They're becoming different things. Yay. Okay. Do we talk about this shit? I don't even remember. I don't think this shit is that important. So, yeah. Cool. So that should be everything that we need to know as of currently. Yeah. Yeah, that should be everything that we need to know for this class. So, alrighty then. I'm pretty much done. So, I hope that helped you guys. If you have something that you don't understand, just tell me and I'll see if I have time to go fix the problem. Wow, look at all these people. It's like, wow. Tons of people studying. Congratulations for those people. Alright, anyways, bye guys. See you next time. Where's the stop recording?